So yesterday I introduced the operon. I'm going to get a little more fancy with it. So the way the operon works, here's a gene, and it has in front of it a couple sites: the promoter, the operator. I'm going to introduce a new site called the cap binding site. Okay? So, if you're a bacterium and you're just hanging around in your normal environment, you don't need to be making enzymes that break down lactose. Lactose is kind of a rare sugar. It's found in milk. But you want to break it down if it's available, but under normal circumstances it's not available. Okay? So you want this gene to be off. So how do you turn it off? Well, there's a regulator gene that when it's transcribed, it codes for a protein that folds into a shape to make what's called a repressor. The repressor bounces around inside the cell until it hits the operator and it sticks to it. I made this too good. Sorry, I'm wrong. It doesn't fit. It needs to fit perfectly for me to be happy. There we go. Okay, so the repressor protein bounces around and it sticks to the operator. Now this gene is off. And most of the genes in cells are off. There's some stuck to the DNA that keeps the RNA polymerase from binding to the promoter and transcribing it. So this gene's in the off position. And like we said yesterday, if you put the bacteria in some milk, all of a sudden lactose sugar will come into the bacterium and one of them will stick to the to the repressor, and it changes shape and falls off, and now the gene's on. So the gene turns on when there's lactose around. That makes sense. Now we can transcribe the gene, RNA polymerase can bind, transcribe the gene, and that'll make proteins like this one, lactase, that will break down lactose and kind of digest it, and eventually it'll break down all the lactose, and when the lactose is all gone, it'll even break down the lactose that's stuck to the repressor. And when it breaks that down, the repressor will change shape back into a shape that'll stick to the operator and turn it on. So the bacteria, the E. coli, doesn't have to think. The gene is turned on when it needs it to be on, and the gene is turned off when it needs it to be off. That was all reviewed from yesterday. Are we good with that? Oh, no. Thumbs up. Okay. Does anyone have a question about this? We're good? Is this blowing your mind, Ashton? You ever heard anything like this? It's crazy. Now, let me tell you about this cap binding site. When the amount of DNA in a cell is low, I'm sorry, not the amount of DNA, when the amount of sugar in a cell is low, then you're going to start, if you're the E. coli cell, you start running out of ATP. And do y'all remember what ATP is converted to when it's broken down? ATP gets converted to what two things? ADP and P. Remember that? Now, if the cell's really low on energy, after it runs out of ATP, it'll start using ADP. And it'll pull that last phosphate off of ADP. Are you with me, Matt? You don't seem like you're with me. And the cell can use ADP for energy under extreme circumstances. Guess what the ADP breaks down into? AMP and P. And this AMP can form a molecule called cyclic AMP. And that's when there's a bond formed between that last phosphate and the sugar. It just forms a little bond, kind of bonds with itself, and makes kind of a circular formation, a triangular formation. It's called cyclic AMP. So, when the cell is really low in energy, you get a bunch of cyclic AMP floating around. This means the cell needs energy bad. And this cyclic AMP will float around. Here's another one of it. And it will attach to a special protein that was created on another gene called CAP. The cyclic AMP attaches to the CAP and forms what's called the CAP-CAMP complex. CAMP for cyclic AMP and CAP for the CAP protein. So when a cell's really low in energy, you get cyclic AMP, it attaches to the CAP protein. This CAP-CAMP complex will attach to the CAP binding site. That sits in front of the Lac, that's at the beginning of what we call the Lac Operon. Now when that happens, it makes this whole area kind of bulge out a bit. It makes the promoter, when this thing binds, watch this y'all, when this thing binds to the CAP binding site, up, the promoter bends. It bends outward like this. And this side, so it's gonna, this thing's going to be stuck out like this. It's kind of coming out at you a little bit like 3D. Again, this is the promoter. And what that does is it makes it easier for the RNA polymerase to attach to the promoter because it's stuck out so much. And what this is, this CAP binding site, what it is, it's like a volume control. If we really want to turn up the making of this protein, the CAP binding, this CAP-CAMP complex will stick to it. This thing will stick out. And then if the cell finds itself in a lactose environment, the lactose will stick here. It'll fall off. And the RNA polymerase will really be attaching to this very well. And you can really make genes that will break down lactose. And the net result of that is, when we break down lactose, we make sugars that can be broken down to make ATP. So we were running out of ATP, that's why the CAMP appeared, and that causes us to break down sugars faster and make more ATP. We basically turned up the volume on this whole thing. So we've got two things here. Binding at the CAP site makes it go faster. Binding at the operon, at the operator, turns the gene off. Isn't that neat? All without the bacteria having the thing. Did I lose everyone?
Yep.
Ask a question.
So does it happen before it attaches to the operator?
Does what happen before what attaches?
The CAM.
This can happen at any time. This can attach to the CAP binding site. Now, it won't do any good if the repressor is attached to the operator. But when the bacteria finds itself in an environment that has lactose, that will be helpful. Because the lactose will bind, and this thing will fall off, and now we will very quickly be able to attach the RNA polymerase and do the transcription.
So if there's no lactose, nothing will happen?
If there's no lactose, nothing will happen. It'll still be off.
Even
if that's bound, yeah.
And that's ATP? That connects to it?
This is cyclic AMP, which is formed from ADP. When ADP is broken down, you get the AMP. and ATP is broken down to make ADP. So you only form AMP, cyclic AMP, when you're really low on ATP, and you need more ATP at that point. Other questions? No? Okay. Now let me talk about the Tryp operon. Are you ready to get trippy?
Mm-hmm.
It's basically the same as the Lac Operon, only backwards. So, we call the Lac Operon an Inducible Operon. Why do we call it an Inducible Operon? Do you know what the word to induce means? Have you ever heard that word?
Mm-hmm.
To induce means to make something happen. Okay? When lactose is present, it sticks to this repressor protein and turns the gene on. It induces the translation of a protein. That's why it's called an inducible operon. You want me to write that word for you? An inducible operon. The Lac operon is an inducible operon. And that means the gene turns on in the presence. How do you spell presence? Is that right?
Sure. Yeah.
Of a molecule. Yes?
Is the inducible operon only used with lactose?
No, there's a similar operon for all 10,000 genes in the prokaryote.
Inducible too?
Some of them are inducible and some of them are repressible. I'll tell you about the next one is repressible. The Tryp operon is a repressible operon. What does that mean? Well, let me explain the Tryp operon to you. So, y'all remember our proteins, right? A protein is a chain of amino acids. Are y'all with me? Gotta pay attention here. A protein is a chain of amino acids. Well, one of the amino acids in proteins is called tryptophan. You may have heard of tryptophan before. They say turkey has a lot of tryptophan and it makes you sleepy. Have you ever heard that? I don't think that's actually true. But anyway, they say it. And, you know, if people say it, then it's probably true, right? Okay, so tryptophan is necessary for making proteins. I got a bunch of tryptophan here. Now, there are genes on the bacterial chromosome that code for making enzymes that build tryptophan. And in the book, it shows this gene. Let me show you the Tryp operon. Here's the Tryp operon. So the Tryp operon also has a promoter. Let's not worry about the cat binding site with the Tryp operon. But there's also a promoter. It's funny the way that sticks out like that. Doesn't do that necessarily. But I like the drawing. Yeah, okay. The promoter. Okay. So here's the Tryp operon, and notice there's 5 genes. Tryp E, Tryp D, Tryp C, Tryp B, Tryp A. Those 5 genes code for 5 different proteins that can build tryptophan. Those 5 enzymes can build tryptophan. Do you want to be building tryptophan if there's a lot of tryptophan already in the cell?
No.
You would want the gene to be off, right?
Correct.
So here's how the Tryp operon works. There's a repressor protein that the tryptophan will stick to the repressor protein, and that makes it into a shape where it will bind to the operator and turn it off. If tryptophan is present, it binds to the repressor protein and turns it into a shape that turns the gene on. So this is the opposite of the lac. In the Lac Operon, when the lactose stuck to the repressor, it fell off. In the Tryp Operon, when the tryptophan sticks to the repressor, that makes it stick, not fall off.
It makes it keep going.
It makes it stick to the operator, and that shuts off the gene. The gene is now off. We have tryptophan around. We're not going to transcribe and translate proteins to make more tryptophan. Now watch what happens as the cell goes through its metabolism. If the cell's making proteins, it's going to slowly use up this tryptophan to make proteins at the ribosome. Remember when we did translation? We're using up the tryptophan, y'all. We're using up the tryptophan. We're using up the tryptophan. We're almost out of tryptophan. Is there any more tryptophan left in the cell anywhere? Yeah, there's one more. There's one more. I'm going to take the last tryptophan to make a protein. And guess what? This thing changes shape, falls off the operator, and now we're out of tryptophan. The RNA polymerase will attach to a promoter, transcribe these genes, and will make proteins that will make more tryptophan.
Wow.
So it's a system. It's called a repressible operon, that when you have a lot of a molecule, the gene turns off in the presence of a molecule. So when you have a lot of tryptophan, it attaches here, gene is off. When you have hardly any tryptophan, it pulls that away, gene turns on. So the tryptophan, the Tryp Operon is kind of opposite of the Lac Operon.
And all those bind together to make more?
All of these,
Yeah.
they're used for making proteins
Okay.
in the ribosome. So these get used up. Now when you transcribe this gene, when this thing sticks and opens this up, it makes proteins. Those proteins that are made can make more tryptophan. They can make tryptophan from scratch. So these proteins here in the Tryp Operon make tryptophan. You don't want to be making tryptophan if you have a lot of it around. Does that make sense? Would you like to see video footage?
Heck yeah.
Oh, dang it.
All right, video footage.
Said that last time we got it wrong.
Video footage of the Tryp Operon.
Writing's not that easy, but Grammarly can help.
Whoa.
This sentence is grammatically correct.
Get back.
The tryptophan operon, Tryp Operon for short, is a bacterial system for regulating the synthesis of the amino acid
Pay
tryptophan.
attention, please.
The operon controls expression of 5 genes. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. But when the cell has lots of tryptophan, why waste energy making more? The Tryp Operon is at one position on the circular bacterial chromosome, and a regulatory gene for the system is about a quarter of the way around the chromosome from it. This gene, called TRPR, encodes a dimeric repressor protein on a slow but ongoing basis, and the repressors diffuse through the cytoplasm. We'll see in a little bit how the repressors work. Now let's look at the control sequences for the Tryp Operon. They include a promoter, the binding site for RNA polymerase, an operator, which overlaps the promoter, and a leader gene, TRPL, which has an attenuator sequence, which we'll also look at soon. When the bacterial cell's tryptophan levels are low, RNA polymerase can bind the promoter and transcribe the structural genes. Each gene encodes a protein chain for an enzyme in the TRP synthesis pathway. TRPE and D for anthranilate synthase, TRPC for indole glycerol phosphate synthase, and TRPB and A for tryptophan synthase. As tryptophan is synthesized, it diffuses through the cytoplasm. When it encounters a TRP repressor, it binds a site on the repressor, changing that protein shape. The repressor can now bind the operator, block polymerase binding, and prevent transcription of the structural genes. Thus, tryptophan is said to be a co-repressor. Like the Lac repressor, the TRP repressor does not stick like glue, but periodically dissociates, allowing a low basal rate of transcription. The operon can fine-tune tryptophan synthesis. In essence, it can read various levels of tryptophan concentration in the cell. It uses a system called attenuation to do this. When the tryptophan level is low, ribosomes translating the leader mRNA as it emerges from the RNA polymerase pause briefly when they encounter two tryptophan codons in a region called 1. They wait for TRP-charged tRNAs in order to continue. And while they wait, a pair of complementary regions of the mRNA, called 2 and 3, form a stem and loop. The next region that could have paired with 3 is not yet transcribed. When the TRP tRNAs appear, translation continues through the structural genes, and thus more tryptophan is produced. If, however, tryptophan-charged tRNAs are plentiful, there is lots of tryptophan in the cell, the ribosomes read through region 1 quickly, which allows region 3 and newly transcribed 4 to pair. This stem and loop terminates transcription, and the structural genes are not transcribed. Tryptophan is not produced, and its concentration in the cell decreases.
They actually got it's more involved than I explained, and they went into it at the second half of that video there. All you need to know for the AP exam is this first part. When tryptophan attaches to the repressor, then it can stick to the operator. That's the TRP operon. It's opposite of the Lac operon. In the Lac operon, this is the Lac operon. When lactose attaches to the repressor, here's the repressor stuck to the operator. When lactose attaches to the repressor will fall off of the operator. That's the Lac operon. So kids have confused the two operons. And the only difference is in the Lac operon, the molecule makes it fall off. In the TRP operon, the molecule makes it stick. That's really the major difference between the two.
Isn't there another one? Like an actor?
Not that you have to know.
Okay.
I don't think. And then the cap binding site here kind of attenuates the whole thing. Okay? So I didn't quite get to that's the end of 16.1 and 2. We're supposed to do 16.3 today. I kind of fell behind because I wanted to do the lab with you. But that's okay. We're going to do 16.3 and 4 tomorrow. So you could get a quiz on 16.1 and 2 tomorrow before we finish 16.3.
But not 16.3 tomorrow?
Not 16.3 tomorrow. That would be the next day. We're a day behind right now. Is that cool?
That's cool.
I did that because Ashton was visiting. I was like, I'm going to slow down a little bit. I don't want to blow this guy's mind. All right, peace out.